Okay, thank you very much for the introduction and thank you for the invitation. I'm delighted to come along and speak to you today. So I am an exercise scientist and I deal primarily with humans. So I hope that I will be able to give you some perspectives from the human research on this topic that will translate to pet care. So if I first of all give you a little bit of a background on the exercise stress model that we use, and give you a little bit of information about where this comes from and basically how that gives us a useful model that we can use and that which translates to clinical situations and clinical settings. And basically this comes from the background where we work with human athletes or individuals with very high physical activity levels, either as part of recreation or as part of their occupation. And what we see is in some of these groups of individuals, they do, some of them tend to pick up illnesses more often than normal. And we refer to these individuals as illness prone, and in particular, illnesses of the upper respiratory tract. So upper respiratory tract infections, or URTIs, as the abbreviation that you can see up at the top of this screen here. And we look at this in two different ways. We use what we call a chronic model, and we also use an acute model. So the chronic model is basically where we will monitor these individuals over several weeks, or more commonly, over several months. And the reason that we use this chronic model is because there are lots of contributing factors in here, but primarily they are all related in one way or another to stress of some description. So whether that's physical stress, psychological stress, environmental stress, or some other form of stress. And it's all of these factors that we're calling the contributing risk factors here, which contribute to this increased presentation of these illnesses. And monitoring these individuals long-term allows us to study the effects of various things and the effects of various interventions at potentially mediating these outcomes. So we have a model here where we can use free living human participants in order to better study and understand risk factors related to illness. We then also have the acute model, and the acute model tends to focus more on the physical and physiological stress pathway. And in this acute model, we would normally have a controlled single stress event. And this single stress event is normally physical exercise. And this physical exercise we would normally have in a controlled setting within a laboratory. And the reason why we use this model and why this model is of interest to us and actually translates beyond humans and across all species really, is because the short-term, the acute physiological stress responses do share many similarities with the responses that you get to more clinical stress situations, such as surgery or trauma. And because of this, we can study these in a controlled laboratory environment. We can look at interventions, and this can help us understand the value or the usefulness of these interventions that potentially translates to these other stress situations, the more clinical settings such as surgery, trauma, etc., etc. So some of the responses that might be useful here are things like we do see trafficking of many immune cells. We do see increases in stress hormones and many immune regulatory factors. And obviously by controlling these with a single stress event, we can get really great insight into the usefulness of various interventions in this context. And we can, of course, use nutrition or study the effects of nutritional supplements on mediating some of these outcomes and some of these responses. So if we're using the chronic model here, this generally tends to be healthy individuals in free living conditions, generally larger scale studies, so sometimes dozens or hundreds of participants, essentially studied in their natural environment. And we tend to focus on immune markers and also illness presentation and look at the impact of nutritional interventions, so nutritional supplements, for example, at actually mediating these outcomes. So this has a lot of advantages because it's applicable to the real world situation. But of course, the limitation is there are so many contributing factors, as we saw on the previous slide, that it is hard to pinpoint a single causative factor. At the other end of the spectrum, with the acute model, these tend to be a little bit more invasive and generally smaller scale because they're lab-based and a bit more demanding of participants' time, effort, etc., and demanding on resources. But they tend to be a bit more insightful in terms of the mechanistic insight that they can provide. So we will commonly look at some of these acute physiological stress responses that I spoke about on the previous slide, and also some of the immune responses, and then perhaps the presentation of illness in the weeks or months after that single stress event. So on the topic of nutritional supplements, obviously what I'm going to talk about today is bovine colostrum as a potential nutritional countermeasure that we have investigated and examined using this physical stress model. And as a quick reminder for those of you who need it, but I'm sure many of you don't, bovine colostrum is the initial milk, and we're usually talking about the milkings or the collected within the first 48 hours postpartum. There are various types, so there is something called hyperimmune colostrum, but I'm not going to talk about that today. This is where the cattle will have been vaccinated against a particular pathogen, and therefore antibodies to that pathogen will be enriched in the colostrum. But I'm just going to talk about normal colostrum from normal cattle. So obviously, if you are talking about a baby animal, so if you are talking about a neonate, then of course colostrum is vitally important in the first few days of life. So it does contain large immune factors, antibodies, immunoglobulins, and so on, and is responsible for the passive transfer of immunity to the neonate. However, that pathway simply isn't open in an adult mammal, and therefore, when we're talking about adults or more mature animals consuming bovine colostrum, it is not this pathway, and we really need to be clear on this, that it is not this pathway that is being investigated, and it is not this pathway that is responsible for any of the beneficial effects that we might see when we're talking about colostrum feeding to older animals. And in fact, there's probably two other pathways or two other mechanisms that are responsible for this, which are primarily related to other bioactive and potentially bioavailable constituents that we find in colostrum. We actually find it in mature milk as well, of course, but it is just the concentration is significantly higher in colostrum. And these two potential pathways or these two potential mechanisms you can see up on the screen here. So the first one is that it's not the larger, passive transfer products, so the large antibodies, for example, that would pass across the gut in a neonate, because they are large proteins and they will be broken down in the process of digestion after you consume this orally. So it's potentially smaller components that are available and are in this product, and that actually do survive digestion and then appear in the systemic circulation and can have these effects within the body. So that's one possibility, that there's really small components that are not degraded or are not massively influenced by the process of digestion and become bioavailable. Or another possibility, or more likely a combination of these two things, is that they are breakdown products, or metabolites of the larger molecules that are broken down during the process of digestion that become available into the body as part of the metabolic breakdown of these products. And regardless of whether it is, these smaller products that are already there or breakdown metabolites, it is likely that when they do get into the body, they then start having biological effects within the body. So this would primarily, if we're talking about immune function and defense mechanisms, this would primarily be things like priming and activating the immune cells, basically enhancing their functioning and therefore strengthening the immune defenses. Another potential pathway is via direct effects of some of these substances and potentially some of the larger growth factors and so on now, actually having a direct effect in the gut by interacting with the cells of the gut lining. And there's a couple of things that can happen here. So what they will do here is upregulate the natural protection and repair mechanisms of those gut epithelial cells, which helps to maintain the barrier function of the gastrointestinal tract. And basically what that does is helps prevent things getting across that barrier that shouldn't be getting across that barrier. I'll talk about this in a little bit more detail a little bit later, but that could have direct effects on the gut and some potential gut symptoms, and also preventing the sort of unwanted passage of components across the gut barrier can also, well, if those things cross the gut barrier, that can also activate the immune system. And basically this is, I guess, using up resource from the immune system. And therefore that could be an indirect pathway by preventing this, via which we can also have an effect on immune function. Okay, then, so if I just talk and give a few examples of some of the studies that have been done in humans who have consumed bovine colostrum as a supplement for several weeks. And there's been a number of different studies on this. And the outcome markers and the outcome measures that have been investigated are really quite many. So on the left of the slide here, you can see there's been some studies that have focused on mucosal immunity. And probably the most commonly studied factor of mucosal immunity is immunoglobulin A, IgA. And for those of you that need a quick overview or a recap of what immunoglobulin A is, essentially this is a first-line defense factor that we have in mucosal secretions, and it helps to protect these mucosal surfaces. It helps to neutralize pathogens such as viruses and bacteria. It helps to prevent them from attaching to mucosal surfaces, and it helps to prevent them from replicating. So it is part of the innate immune defense. And generally speaking, higher levels of this in mucosal secretions would tend to indicate a better defense via this particular mechanism. And what we've seen in human studies here is that individuals who take daily supplementation with bovine colostrum for several weeks have shown an increase in this marker in mucosal secretions such as saliva. So that's what the top graph there shows, was a study with 12 weeks of daily supplementation in a group of endurance runners. And basically the group that had the bovine colostrum supplementation saw a significant increase in this particular immune marker. There's been some other studies, so the reference you can see at the bottom of the screen there. Again, they saw some beneficial effects on salivary levels of lysozyme, which is an antimicrobial peptide, which is quite important in innate immune defenses. And this was with 20 grams per day for 4 weeks. Other aspects of immune function, so if we now look at the functioning of immune cells and how well they respond to stimulation and basically how effectively they do their job, there's been some research showing that they are also benefited in individuals who have consumed daily bovine colostrum. So again, the Davison and Diamond study here is the same study as the one referenced on the bottom left of the screen. And in this study, there was an acute stressor, which was 2 hours of moderate intensity exercise. And generally speaking, we see a decrease in some of these cellular markers of immune function. And there was protection against that decrease in the individuals who had consumed the bovine colostrum. And this particular cellular function was neutrophil response to stimulation. So the neutrophil being the most abundant white blood cell, and obviously is very important in both innate immunity, but also adaptive immunity. And there've been a number of studies showing with in vitro addition of bovine colostrum that the neutrophils are primed and their function is enhanced. So it seems to support the idea that there are constituents and components within the colostrum that become bioavailable, and they can have various effects throughout the body on immune defenses and on immune cell functions. However, having said that, there are also quite a lot of studies that don't see these benefits in individuals who consume this product. There are potentially a number of reasons for this. So one of the reasons is probably down to the particular product used. So there is large variability between the quality of products. And research that has tried to identify the bioactivity of different colostrum products has shown that some have very, very low activity and some have very, very high activity. So basically, the quality of product is very important. And we need to make sure that we are using a product that has high bioavailability and high bioactivity. But also, a lot of these studies have focused on single markers or just a few markers that are really obtained in, blood samples or saliva samples. And basically what we're doing there is we're taking the immune cells or the immune factors outside of their natural environment. And it is generally considered that although these might be useful as sort of supplementary and additional markers, they're not really the strongest markers in their own right, because they're just measuring one very small component. So the pyramid that you can see on the left of the screen there is coming from the Albers reference. And basically what that acknowledges is that some of these isolated markers in blood and other fluids on their own are not the highest value. And what we're really interested in, or what we really should be interested in, is the clinical presentation, clinical symptoms, and also clinically relevant immune responses or clinically relevant markers, which give a representation of the whole integrated immune system working together as a functional unit. And what is generally well accepted is that markers which challenge the immune system and quantify this in an in vivo way are generally considered higher value. So again, referring back to the pyramid, challenge tests, which basically don't just look at isolated markers, but they look at the integrated functioning of the whole immune system working together in a coordinated way, are generally considered of higher value. And with our exercise stress model, we have implemented such a test, which we would consider to be of clinical relevance and gives a good representation of this integrated response, which is considered high value. And the model that we've used here, again, is the exercise stress model, whereby we have individuals undertaking a physical stressor. So this is 2 hours of treadmill running in this particular example. And before that single stress event, so this is an acute stressor, they were consuming daily bovine colostrum supplements at 20 grams per day for 4 weeks. And the challenge test that we use is we basically sensitize them to a novel antigen. So this is something that they have never encountered before, their immune system has never encountered before, and we expose them to it via presentation on the surface of the skin. And the picture that you can see in the top there with the little circles, that is the upper aspect of, or the inner part of an upper arm in a human. And this is the recall that we do after we've sensitized them. And we basically expose them to this antigen at varying doses. And we can then quantify the magnitude of the immune response to that antigenic challenge. And we can do this in a full dose series so that we can then calculate the sensitivity, i.e. what is the minimum level of this antigen that the immune system can detect and respond to. So we do this acute exercise stress before we sensitize them, and then we test the strength of the immunological memory that is developed to this 4 weeks later. And basically, if we compare a rested individual to somebody who's been through this acute single stress event, then the person who's been through the stress event will generally have a 50% lower sensitivity to this antigenic challenge. So the stress model definitely does reduce this clinically relevant integrated marker of immune functioning. So if we put 2 groups of individuals through this stressor, and if one group has the daily bovine colostrum and one group had a placebo, then we can see whether this supplement actually has a beneficial effect on this clinically relevant marker. And in this particular study here, so Jones et al. In 2019, it did just that. So basically what we're saying here is there was a 50% improvement in the sensitivity to this antigenic challenge, which was exposed at the surface of the skin, to which they were sensitized immediately after a 2-hour treadmill run. So under normal conditions, we would expect a 50% reduction after exercise, but with colostrum, that reduction was significantly prevented. The other thing that we can, of course, look at is clinical presentation itself. So whether or not an individual actually reports with a symptom. And here we're focusing on upper respiratory tract infection once again. And there's been a number of studies where they have examined this. So some of these you can see on the screen now. And in many of these studies, where we quantify the presentation of upper respiratory illness, what we see is a significant, or a tendency for a significant, reduction in the incidence, so the risk of picking up these types of illnesses, in the individuals who had the bovine colostrum supplementation compared to the individuals who had a matched placebo. And generally what we see is in the larger studies, so the studies which are generally more powerful, statistically speaking, we do see significant protection in the individuals who'd consumed bovine colostrum. And this was summarized a few years ago in a meta-analysis, which you can see presented on the screen here. And basically what they quantified here in a meta-analysis, where they pooled the data from 5 smaller trials to get a kind of general consensus on our understanding about this, was that the number of upper respiratory symptom presentations, or the number of episodes, was around about 40% lower in colostrum compared to placebo. And in individuals who did present, the number of days for which they suffered with that particular illness was again around about 40% lower with colostrum supplementation as compared to the placebo condition. So what we have here, when we look at the clinically relevant markers that measure the integrated immune response working together in vivo, and also when we measure these clinical presentations, so whether or not somebody actually presents with an illness, then we do have really robust evidence that bovine colostrum can protect the immune system and protect against the occurrence of these illnesses somewhat. Okay, another area where bovine colostrum has been researched and investigated quite extensively, and we've done a lot of this in my research group in our labs, is the effects on the gut and protecting against intestinal cell damage, and therefore the consequent disruption of the barrier function, and therefore increased permeability, or something that you might colloquially hear referred to as leaky gut. So clinically, the risk for increasing intestinal permeability is that it allows the translocation, it allows the passage of bacteria and or bacterial toxins from the gut into the systemic circulation. And what can happen if, this always happens to a relatively small degree anyway, but it's reasonably controlled, and we normally have very small amount of permeability and a very small amount of these things crossing the gut barrier and getting into the circulation. But if we put individuals under stress, and in some clinical situations, this increased permeability, which we can get as a result of certain stressors and certain insults, can increase this bacterial or bacterial toxin translocation. And we can start to see an increase in this in the systemic circulation, and then we can start to see some endotoxemia. Now, at a moderate level, this could be a contributing factor to, some of the milder symptoms that you can see in the middle box there. So things like nausea, vomiting, cramping, diarrhea. So some general gastrointestinal upset type symptoms that we often see reported in individuals who suffer from this. The rare but more severe outcomes, which are obviously more worrying, are things like you can get a severe activation of the immune system. It can contribute to things like sepsis, shock, and even in extreme situations, multiple organ failure, and maybe a little bit less severe heat illness in some individuals. So again, if we have really significant increase in permeability, there can be some quite serious consequences. So there's been some animal work on this where they have some of the contributing factors here are things like increasing core body temperature, which has relevance to some clinical situations, and also certain medications and various other stresses and insults can cause this damage to the gut cells, and therefore increased increased permeability, or a reduced ability of the barrier to function in the way that it should. And this has been studied in animals, whereby they've heated the animals until they reached a core temperature of around about 42 degrees Celsius, which is very hot. And And this does increase core temperature significantly. But if those animals were put, and this was done in a rodent study, so that top one that you can see there. And however, when the individual animals were provided with supplemental bovine colostrum in their diet, then that damage and that increase in permeability under that heat stress was actually prevented. In humans, we know that if you administer non-steroidal anti-inflammatory drugs, so drugs in this class, we know that they do some nasty things to the gut, and they can cause some damage to the gut cells and increase the gastrointestinal permeability. However, there's been some research showing that if you administer bovine colostrum before and with this class of drugs, then it actually prevents the increase in damage and permeability that this causes to the GI barrier. We've taken this a little bit further and explored this using our exercise stress model. Because with the exercise stress model, again, we do see an increase in gastrointestinal permeability for a short period afterwards. This is useful because it allows us to study again the effectiveness and the utility of various interventions at protecting against this. But the amount of increase in permeability that we see with the exercise stress model is relatively minor compared to these more extreme clinical manifestations. And also it's short term and it's generally safe. But what it does allow us to do is to have very, very tight control over all of the potential extraneous variables and also the dosage of the stressor, so the intensity and the duration of the exercise. And what you can see on the graph at the bottom left here, so the Marchbank study from 2011, you can see to the very left of that graph, there are two bars, and this is an index of gut permeability at rest. So this is what it should be normally, and this is what we typically see at rest. Incidentally, the way that we quantify this is by feeding participants a non-metabolizable mixture of sugars. We have a small sugar and a large sugar. The small sugar will normally pass across the gut, get into the blood, and it will then be removed and excreted in the urine. The large sugar shouldn't pass. So we should have a relatively low ratio of the larger sugar to the smaller sugar in the urine samples that we collect over a 5-hour period afterwards. If we see damage to the gut, and if we see an increase in permeability of the gut, we start to see that more of the large sugar are able to pass into the circulation and then end up in the urine sample. So if we see an increase in the ratio of the large sugar to the small sugar, this tells us that the gastrointestinal permeability has increased or the gut has become more leaky. And that's what we're showing on this graph here. So the two bars to the left in the placebo condition are normal resting samples. The bar to the right in the placebo condition is what happens after exercise. And we see a significant increase in GI permeability as identified via this marker. However, the bars to the right of this graph show the same scenario, but where the participants have had bovine colostrum as opposed to placebo for the 2 weeks before that exercise stressor. And what you can see here is we get a substantial blocking or a substantial blunting in that exercise-induced increase in gastrointestinal permeability. And this is probably caused in part by the protective effects of bovine colostrum on the gut and limiting the amount of damage to the cells of the gut lining and therefore better maintaining that barrier function. So the graph that you can see in the middle there from March and colleagues in 2017, this basically did something very similar. This marker, however, is a marker in blood which is released from cells of the gut when they are damaged. So the higher this marker suggests that the higher amount of damage that has occurred directly to these gut cells. And again, what we can see here, the bars to the left, which are the placebo, they are pre and post exercise. So following exercise, there was a significant increase in damage to the gut cells, but that increase in damage was prevented when the participants had been feeding or had been consuming bovine colostrum for a couple of weeks before this acute stress event. This was 2 weeks of supplementation. In a follow-up study, we did investigate whether or not there was any benefit with only 2 days of supplementation. And that's what you can see in the third set of bars there. So we've got 2 resting baseline samples. We've got the low index of gut permeability. We've got the next one along where they did exercise and there was a significant increase in permeability. And the colostrum for 2 days, this was after 2 days of supplementation, there was no protection there. However, when they continued that supplementation for 14 days, we did then see protection. So what this suggests is that there is a benefit, but that didn't occur with 2 days and you need more than 2 days. We don't know exactly how much or how many days between 2 and 14 from this study, but it's probably, closer to the 2 weeks. So probably 1 to 2 weeks as an absolute minimum. Okay then, so to summarize, I've shown you some studies there using the chronic and acute human exercise stress models. And hopefully you've found that, or you've understood that there is some potential value and potential translation that we can take from these and apply, across species to many clinical situations. First of all, as a dietary supplement, there is good evidence that bovine colostrum has beneficial effects in adults. And of course, this likely translates to many species. It can protect the gut against stress-induced damage and therefore prevent the excessive increase in permeability that we get with certain stressors. It can protect against stress-induced immune disturbance and therefore reduce the susceptibility to illness and infection under acute and chronic stressors. However, it's likely to see the most benefit that we need to do this daily for at least a couple of weeks as a minimum. And importantly, product quality is likely to be very important because we do see variability. Some products don't really have the same benefit as other products. So a really important practical consideration is to use a high quality product from a reputable supplier. Okay then, so thank you very much for your attention. I hope that you found that useful and interesting, and I look forward to answering any questions that you have in the Q&A session later.